BIOL 100, Chapter 38, the musculoskeletal system. Alright, so the whole purpose of having a musculoskeletal system is to cause or control movement, and more specifically, especially in humans, it's to support or maintain an upright posture, allow our bodies to move by transporting our bodies and manipulating objects, as well as to protect our bodies, protect our internal organs, our tissues, everything like that from getting more damaged in different situations. All right, so there's a few different kinds of skeletons that the book talks about. The first is hydrostatic. These are mostly marine invertebrates, which we'll see here in a second with like a sea star. But sea anemones, earthworms, which is not marine, but Cnidaria, those are like jellyfishes and things like that, as well as some other invertebrates. But these guys have a fluid-filled compartment within their body called a coelom. And that compartment is under hydrostatic pressure, or essentially like water pressure, because of the fluid that's in that body cavity, and it supports all the other organs of the organism. We're not going to talk too much about it because we're a human biology class, but just know that this exists. And again, mostly with marine invertebrates, like a sea star. Okay, and then we also have exoskeletons. You've probably heard of exoskeletons talking about different insects or crabs like arthropods. And exo meaning outside, right? Endo meaning inside. So we have an endoskeleton that we'll talk about here in just a second. But an exoskeleton is external, and it's a hard encasement on the surface of the organism that provides defense against predators, also supports the body, and allows for movement through contraction of attached muscles. So crabs and lobsters have this, and theirs is actually made out of a large percentage of chitin, which is strong but flexible. And because that exoskeleton is acellular, they have to periodically shed or molt their exoskeletons. That's why you'll see little like crab exoskeletons as you're walking the beach and you're like, oh my gosh, it's a dead crab, but really it's just a molt of their exoskeleton. So they were not harmed in the process, they just were shedding their skeleton because it doesn't grow with them. So here's an example of a Halloween crab. You can't really actually see all the muscles attached to the exoskeleton, but if you ever held a crab or looked at their molts or anything, it would give you a really good idea of how this is formed and what they're kind of like. Okay, into the main skeleton type that we will focus on in this class, which is an endoskeleton. And similar to an exoskeleton, it's providing support for our body, protecting our internal organs, and again, allowing for movement through contraction of muscles that are attached to it. But we've got hard mineralized structures located within the soft tissue of organisms, or within humans, let's say. And we have a couple of different types of skeletons in the human. So we have the axial. So this has the skull as well as the ossicles of the inner ear. That's what allows for vibrations of the inner ear, and that's how we can hear and sense movement and our, where our body is in space. We also have the hyoid bone here, which is responsible for like tongue movement, and a little bit of the jaw movement as well. Vertebral column, as well as the rib cage. So that's the axial skeleton. Going in a little bit deeper, we have part of the axial skeleton is the skull. So we have bones of the skull that are supporting the face and protecting the brain. We have our different parts of the skull here, or even different lobes of the brain: frontal, parietal, temporal, and occipital. And then you see here we have different parts of the skull as well, like maxilla and mandible. So those are controlling our jaw movements. Lacrimal, nasal, ethmoid, sphenoid. So those are the main components of the skull, along with the cranial bones, including the frontal, parietal, and sphenoid bones. Those are our cranial bones, and then we also have our facial bones over here that you can see, including our maxilla and mandible. And these guys basically are forming the face, and they're providing the cavities that we have for our eyes and our nose and our mouth. The next part of the axial skeleton is our vertebral column. This consists of our cervical vertebrae, our thoracic vertebrae, our lumbar, and our sacral vertebrae as well, or sacrum, as well as our coccyx. Um, we've got about 33 vertebrae that we're born with as as humans, and then as we get older, some of them, like in our sacrum and our coccyx, those actually fuse together as you get older, which is why we kind of shrink as we get older, right? And for each vertebral body here going down the vertebral column, every single one of them has a large hole in the middle. And that's basically what your spinal cord and the nerves associated with that are passing through. And then there's little notches on the side here as well that hold other nerves that are sending messages back and forth between your central nervous system, your brain, and your spinal cord out to your peripheral nervous system into your body to tell your body what needs to move, what skeletal muscles need to do what. You can see that we have a natural curve to our spine, and in some people we see more of you know, a, we have like scoliosis where you have a little bit more of an S to it. Different things that you have more pronounced parts of your curve, or they might be, instead of curved this way, if we're looking at it from the back instead of a side profile, we might still see kind of an S. But the reason why we have this natural curve is because it does increase the strength and flexibility of the spine. It makes it more like a spring. So it gives you a little bit of shock absorption. And we also get that from the intervertebral discs that are in between these vertebrae. And those, as we get older, become a little bit less elastic as well, which is why you can jump around and do everything when you're young. And as you get older, you don't really have that shock absorbing value that you used to have. So doing those things hurts a lot more. It takes a lot more out of you and can potentially be a little bit more damaging than it was when you were younger because you just don't quite have the body that you used to. The last part of our axial skeleton here is our thoracic cage, or commonly referred to as our rib cage. That's what's protecting our heart and lungs. You can see the costal cartilage here, our sternum, which is essentially our breastbone. It's fused of 3 different bones within the adult, and all of that, or our ribs, we have 12 ribs that are connected to that, with the exception of our last 2 ribs, which are actually technically free floating and are not attached to the thoracic cage. But the thoracic cage in general is attached here to our breastbone and this cartilage, and it loops around to the back and attaches to the thoracic vertebral column as well. Moving on to the appendicular skeleton. So this is essentially the, well, I want to say the lower half of the skeleton, but really it's everything that wasn't part of the axial skeleton that we talked about, which was the skull and the rib cage, thoracic cage, and vertebral column. Now we're looking at our upper limbs, our lower limbs, as well as our pectoral and pelvic girdles. So pectoral limbs would be your arm, forearm, and hand. Pelvic limbs would be your thigh, leg, and foot. And the pectoral girdle here up on top is also known as the shoulder girdle, and the pelvic girdle on the bottom. Okay, so starting with the upper limbs, these consist of the humerus of the upper arm, which is the largest bone in the upper limbs. Um, you also have the ulna of the forearm and the radius, which is on the thumb side. So when you're taking a pulse, you'll hear it called radial pulse if you're taking it from there. Um, and then you have 8 bones of the carpus, which is getting into the hand, 5 bones of the metacarpus, and then 14 bones of the phalanges. So the phalanges are basically your fingertips. You've got 3 phalanges on each digit, with only 2 being on the thumb. Okay, our pelvic girdle. So this is our bottom half essentially. So pelvic girdle is supporting a lot more weight than, say, our pectoral girdle, which is our shoulder girdle up on top. So you'll see the bones here, as well as in our lower limbs in general, are thicker and more robust than on the upper half because this is our weight supporting half, right? The upper half we want to be strong as well, but it's not like we're walking on our arms. So You'll see that a little bit more in the pelvic girdle as well as the lower limbs. And in addition, you'll see a difference between the male and female anatomy, or not anatomy, sorry, they are the same, but you'll see a difference in how wide and how different they are, which is why you'll see girls with bigger hips and males are a little bit, girls have that hourglass shape that kind of goes out and then back in, and males are kind of just straight. And it's because the pelvic girdle has evolutionarily evolved so that women are able to reproduce successfully. So it's lighter, but it's also wider and shallower and has a broader angle between the pubic bones than the male pelvis does. Okay, lower limbs. Our largest bone here is our femur, which is our thigh bone. We have the kneecap, which is the patella. Our leg has both the tibia and the fibula. Our ankle is made up of tarsals. Then we have metatarsals in the foot, and then phalanges in the toes, again with 3 for each toe. It's a close-up of the bones of the human foot and ankle. So again, the ankle is the tarsals here that are connecting your leg bones into the feet and essentially transferring the weight there. So they do actually need to be pretty strong. And then we have our metatarsals, our 5 bones of the foot here, and the phalanges that we just talked about. Okay, so getting into different types of bone, we have a few different types. We have flat bones, irregular bones, short bones, femurs, and sesamoid bones. So long bones are longer than they are wide. They have a shaft with 2 ends, mostly compact, and these are the bones of our limbs. So our leg bone, our arm bone, our humerus, our um, femur, those are all considered long bones. And then we have our short bones here. So these are cube-like. They're often called cuboidal bones, actually. They're mostly spongy, and you can see them in a lot of times, like things like your foot. We have cuboidal bones kind of on the bottom of the foot. So those are always kind of weird breaks if that happens. But within the short bones too, we also have things called sesamoid bones. These are embedded in tendons, so that's like our patella in our kneecap. We also have flat bones. These are spongy bone embedded within parallel layers of thin, compact bone. And this is usually in places like our breastbone or our ribs, things that need to protect some really important internal organs. And then we also have irregular bones. So these are things like our vertebrae and hip bones that have kind of notches to them. They're a little bit irregularly shaped. They're complicated shapes oftentimes, and usually they're spongy with kind of a thin covering of compact bone. Going into a bit more detail on the long bone here, we have it, again, this is mostly our limbs, our upper and lower limbs, but we have the epiphysis here, the diaphysis here, and the epiphysis on the bottom end. So the epiphysis is the wider section at each end, and it's got more of like the spongy bone and the articular cartilage, and it contains the bone marrow as well. And the diaphysis is the shaft that runs along the long bone in between the epiphysis. So, yeah, sorry, I think I said that the epiphysis has the bone marrow. The diaphysis does. So you can see it here in yellow, and it's usually pretty much filling this cavity here. We also have the red bone marrow, which includes the red blood cells as well. All right, so we're talking about bones. What are bones actually made up of? So we have both compact bone and spongy bone. And in the compact bone, we have these elongated cylinders parallel to the bone long axis. And they are referred to as osteons or even reversion systems. And they have concentric rings, which are known as lamella or lamellae, if we're talking plural here. And those are made up of unidirectional collagen fibers along the long axis, and adjacent lamella have collagen in opposite directions. So it's basically just providing even more structure and support for the bones. And this central haversian canal, which we can see going through here, is a core of the core of the osteon. So it has the blood vessels and the nerve tissue. And then perpendicular canals, which we also refer to as perforating or Volkmann's, those are connecting the periosteum to central and medullary cavities. This has our blood supply and again nerve tissue innervation. And then we have lacunae, which are cavities containing osteocytes, and canaliculi, which connect lacunae to each other and the central canal. So the canaliculi is actually essentially where the blood flows in between all of this within the bone tissue. All right, so getting into intramembranous ossification. So this is essentially how bones are being formed and kind of how they're hardening, right? And keep in mind, this is different than calcification. They both involve similar processes, but it's not just calcification. It goes a step further into ossification as well. So what first happens is the ossification center forms within the fibrous connective tissue membrane, and these mesenchymal cells differentiate into osteoblasts. Then we have bone matrix that's secreted into the membrane, and those osteoblasts secrete osteoids. Then we have the woven bone and periosteum being formed, and there's a network of trabeculi, so remember the spongy bone, enclosing those local blood vessels, and those exterior mesenchyme differentiate into the periosteum. Those trabeculi thicken or spongy bone, and they form a bone collar, replaced by the lamellar bone, and then the spongy bone persists to make that red marrow, which is basically the bone marrow that has the red blood cells in it. So while the compact bone is kind of the outer covering of the bone and provides a little bit more strength and support, the spongy bone is within that, right? So this doesn't have osteons and it's organized seemingly irregularly. It's a little bit less organized than we saw with the osteons or the perversion systems in the compact bone. And this does not have osteons. The spongy bone has something known as trabeculi. And these are needle-like or flat pieces. And again, they appear less organized than structures within the compact bone. And it's because they're designed to handle stresses coming from different areas. So you have parts of it that are designed for stresses coming a certain way within your body. And this is kind of allowing for absorption of those stresses. You can see here the trabeculi within the spongy bone are arranged such that one side of the bone is going to bear the tension and the other is going to withstand compression so that they are not easy to break. So you can see the tension lines here as well as the compression lines. So they're kind of going back and forth depending on what bone we're looking at and how it's normally taking stresses. Okay, getting into endochondral ossification. So this is where we're using that cartilage bone as a pattern for our bone construction. So the primary ossification center is at the center of that hyaline cartilage, and that's broken down during the ossification. And prior to ossification, we have that perichondrium is getting infiltrated by blood vessels and becoming the periosteum. And then all of the underlying mesenchymal cells are differentiating or becoming basically osteoblasts. Then we have ossification actually happening where that bone collar forms hyaline model, and then the cartilage in the center of the diaphysis calcifies. The periosteal bud invades the internal cavities, which brings in the blood vessels, nerve tissue, lymph tissue, osteoblasts, and osteoclasts. And those osteoblasts then secrete osteoid around the remaining hyaline cartilage. And that's how we're continuing to ossify it. We're forming that medullary cavity, and the epiphysis also ossify. So you can see it kind of described here visually. Starting with that hyaline cartilage model, moving our way until we have that primary ossification center. Then we have the periosteum, which is covering that compact bone. Our secondary ossification center you can see up here, but now we've integrated the medullary cavity where we have the artery and veins that are providing nutrients to the bone. And then we get the spongy bone here in between the articular cartilage. So that compact bone and then the spongy bone here. And you can see the compact bone on the outside, and we can see the epiphyseal plate here separating it. So it's just the process of how your bones are developing from hyaline cartilage. We start this development after about 6 weeks of fertilizing the egg. So it happens pretty early on, but as you know, your bones calcify as you get older as well. That's why, like, when you have newborns, their bones are pretty soft and they're very moldable. So you have to be really careful with them. But the periosteum here is just connective tissue on the outside of the bone that it's essentially acting as the interface between the bone and all the blood vessels, tendons, and ligaments as well. Okay, so getting into different joints that we have within the human body or within other vertebrates as well, but we'll specifically talk about humans, is we have these fibrous joints. So we have sutures, syndesmoses, and gomphoses. So sutures are essentially the joints that are within the skull. So there's very limited movement here, right? We don't want these plates moving around very much. And they're held together by this fibrous connective tissue. The syndesmoses are good examples are like in between your foot and your ankle, your tibia and your ankle, where you're having a little bit more movement. And then the gomphoses are actually in between your teeth and the socket. Up next, we have cartilaginous joints. So these are joints in which the bones are connected by cartilage. We have synchondrosis and symphyses. Synchondrosis, the bones are joined by hyaline cartilage. So these are found in epiphyseal plates of growing bones in children, whereas the symphyses, these are hyaline cartilage covers at the end of the bone, but the connection between the bones occurs through the fibrocartilage. So symphyses are found at the joints between vertebrae, and either of these cartilage types, you're not going to see very little movement, right? We don't want to have these joints moving too much, or we could be dislocating or breaking different parts of our body as well. You can see the gomphoses that I was talking about, or the gomphosis that I was talking about in the last slide right here as well. Up last, we have our synovial joints. These are the only joints that have a space, or what is referred to as a synovial cavity within the joint, and that's filled with synovial fluid, which essentially is lubricating the joint. So we have different types of synovial joints. There's 6 different types, and they depend on what kind of movement they allow, essentially, and there's different classification of joints on the basis of function, right, that we just went over. So you have like synarthrosis, which is any joints that are movable, which would be like our sutures, gomphosis, and synchondrosis, which we talked about. Amphiarthrosis, which are joints that allow slight movement, so our syndesmosis and our symphysis, and then our diarthrosis, which allow for free movement of the joint, and that's like our synovial joints. So now talking about the different kinds of movement we have gliding movement, angular movement, rotational movement, and special movements. So all of that usually kind of falls into synovial joints, which are the diarthrosis joints that have free movement. So within those, we have different kinds, right? We have planar, hinge, pivot, condyloid, saddle, ball and socket, and each one kind of has different functions that it allows for, right? So you can see the pivot joint here. The example is between C1 and C2 vertebrae. Ball and socket, that's like our hip. Condyloid, this one is like between the radius and carpal bones of the wrist. Plane joint here, or planar joint, between the tarsal bones. Saddle joint here between the trapezium, carpal bone, and first metacarpal bone. And then a hinge point, which is like your elbow or knee. So types of synovial joints, just to give you a better example, or a closer picture, I should say, not really a better example, but the joints of the carpal bones in the wrist are a good example of planar joints. An elbow joint, again, is a good example of a hinge point. You can see the ulna and the radius here connecting to your elbow joint that connects it to your bone in the arm, which is the humerus, that really large bone. And then the joint in the neck that allows your head to move back and forth is also an example, or sorry, not also, but is an example of a pivot point. The metacarpal phalangeal joints in the finger are example of condyloid joints. And then moving on to a different kind of synovial joint, we have the carpometacarpal joints within the thumb. Those are example of saddle joints. You know, we have the opposable thumbs, which is different, classifies us as different as other animals, and that's largely due to this kind of saddle joint. And then we have the shoulder joint, which is a good example of a ball and socket joint. Other example would be like your hip. So you can see here the clavicle, which is that really delicate bone kind of up on top of your chest, connecting your pectoral girdle, the scapula here. Oops. The scapula here connecting to your large arm bone, the humerus. So this is like a ball and socket. But going into the muscular tissue, we have 3 different types of muscle tissue that we've already talked about a little bit, but we'll go a little bit deeper here now as well. We have skeletal, smooth, and cardiac. And again, smooth muscle cells are shorter and they're tapered at each end. They only have 1 plump nucleus each. These are not striated, whereas the skeletal and cardiac muscles are striated, and we'll see why here in just a second. Here's a skeletal muscle fiber. So you can see it's surrounded by a plasma membrane called the sarcolemma, and a cytoplasm is the sarcoplasm within the muscle fiber. A muscle fiber is basically just a muscle cell, and within the muscle fiber, we have many different fibrils. Sorry, I should say the fibrils are really cells. So within a muscle fiber, we have tons of myofibrils within it that are packaged into nice orderly units. And each one of those fibrils has what's called a sarcomere within it as well. So a sarcomere is in between this thing called a Z line and this other Z line. And then there's filaments that are making up the microfibers, or the myofibers. And they're composed of both thin filaments called actin and thick filaments called myosin. I put up a video that I recommend that you watch by Amoeba Sisters, because they're just kind of fun to watch. And I think they do a really good job of animating these things. And they say the way they remember which one's which is actin is pretty close to thin. So actin is our thin one, myosin is our thick one. So we have the M line here, and that's kind of separating these fibers, and we'll see what happens with it in just a second, how that all comes together. But remember, our Z line to Z line, that makes up 1 sarcomere. So how do muscles actually contract? Well, it is by this thing called a sliding filament model. And what we have here is we have our sarcomere, right? And when that contracts, what actually happens is it doesn't get shorter, but our filaments slide past each other. So our actin will slide past our myosin, and our Z lines move closer together. So our A band here in the middle, our sarcomere basically, stays the same length or width, but our I band right here, the space between the myosin filaments, so you can see right here to right here, across this Z line, this will get shorter as these contract. So you can see how it's shorter here and it contracted, and the A band has stayed about the same width. So what happens is it forms a cross bridge, and that's how the muscle actually contracts. This I don't want to explain too much here because I think you really need the animation. There are also a couple animations in your reading, and I would recommend watching those as well, but the Amoeba Sisters one that's posted on the Lima is really great. But what happens basically is there is this molecule here, and or sorry, on actin there's a molecule called troponin and tropomyosin. So basically what this does is it prevents myosin heads, which are like what wants to link onto here to make it contract. It prevents those from actually being able to link. So when a neural signal goes to that muscle that says, hey, I want to move my bicep, then it's going to send a neural signal down there. It's going to release all of this calcium into the cell. And then that calcium is what's actually going to bind to the troponin. And that's going to make it so that the tropomyosin essentially kind of like falls off, not really, but kind of comes off the actin. And it allows the myosin head to form this cross bridge with the actin. When this power stroke happens, what happens is the myosin bends. So it's kind of moving towards the left, you see here. And in that process, our myosin head that used to have ATP on it, which is adenosine triphosphate, it's the body's energy currency, right? If we want to expend energy, we have to use what's called ATP. So the energy currency here, think of it like cash, right? If I want to go to 7-Eleven and buy something, I have to give them cash to buy it. Same thing here. If I want to expend energy, I've got to give that cash up, which is the ATP. But when that myosin head with ATP links onto the actin, it's going to bend this way, and the ATP is going to come off as ADP and a phosphate group. So ATP is adenosine triphosphate, tri meaning 3 phosphates. So it's going to break up into ADP, adenosine diphosphate, and a phosphate group. And then after that, a new molecule of ATP will attach to the myosin head, causing the cross bridge to detach. And then the ATP hydrolyzes the ADP and phosphate, which returns the myosin into its cocked position so that it's ready to go through this process again. We just got to make sure that we have the calcium so that it can actually bind to the actin and go through that whole contractile process. Again, I know it's kind of confusing. Make sure that you watch a video on it. It makes a lot more sense if you can see it animated. And the way that this is happening is because we're receiving neural signals that then lead to action potentials. We'll talk more about action potentials as we start getting into the nervous system, but basically what happens is we have this neurotransmitter called acetylcholine, and it's being released from axon terminal that's binding to receptors on the sarcolemma, or the plasma membrane basically of a muscle cell. And then that action potential is generated and it's traveling down what's called a T-tubule. So you can see the T-tubule here, that's within the muscle fiber. And that's essentially just kind of like the canal that's getting the calcium into the muscle tissue. Okay, so it's going to be released from the sarcoplasmic reticulum in response to that change in voltage that we had up here with the acetylcholine. That calcium, again, is going to bind to the troponin, and that is going to release that tropomyosin so that the cross bridge can form between our actin, our thin filament, the blue one here, and our myosin here, the pink one. Once that happens, acetylcholinesterase removes acetylcholine from the synaptic cleft. So that's happening up here where our neural signals are reaching our muscle to tell it, hey, here's what you're going to do. And then calcium is transported back into the sarcoplasmic reticulum, and tropomyosin binds the active sites on actin, which causes the cross bridge to then detach. So it goes in a full cycle, and this happens every time that we contract our muscles. So it sounds like a lot, and it is, but it's all happening very, very quickly. If you think about all of these voluntary movements that you're doing with your muscles, remember skeletal muscle is striated, and it's striated because it has these actin and myosin filaments. That's what makes it look kind of striped or striated. And these are all of our voluntary movements. Remember, cardiac muscle also is striated. It looks striped like this, but it is not a voluntary muscle. We do not control our heart rate. So that's a little bit on the types of muscle that we have and how they're contracting. Again, please make sure that you watch those extra videos, both in your reading and the one posted on La Lima, to better understand especially muscle contraction and even the difference between the muscles. We've gone over skeletal versus smooth and cardiac in another lecture as well, but this should give you a really well-rounded idea of the musculoskeletal system so that you can then go on to research and discuss different disorders that could happen within the system.